So thank you for joining us this morning. Marco is our speaker today, so he's a BrIAS fellow. So he's got his master degree in plant molecular biology at Ferrara University. So I learned that it is in the Bologna region. In 2018, he got a PhD in evolutionary biology and ecology. The topic of his thesis was on the transcriptional regulation of proline synthesis in rice. He worked a little bit under the sun in the equator in plant protection against banana. Then he came back to Paris. He worked at Agro-Paris Tech Versailles on elicitor properties of bile acid, I guess on plants. So then he worked with David on a MIS project on the characterization of elicitor properties of oxidized sugars from LPMO. So that's the topic of the talk today. And since October 2021, he's a postdoctoral fellow from the FNRS, so the Fonds pour la Recherche Scientifique in Belgium. Fun fact about Marco, what kind of instrument do you think he's playing? Trumpet, flutes? No?
Triangle.
Piano. Drum, he's playing drum. And if you have the opportunity on the 21st of May in Anderlecht, Marco will perform with his group, Modotti, M-O-D-O-T-T-I.
Yes,
it's going to be in Anderlecht Atelier de la Scénographie on the 21st of May. With this, Marco, the floor is yours after a little bit of advertisement. So, is it sharing? I think so.
Okay.
And as always, if you have questions, you just write them in the chat. We have very few people online, so you can also unmute your microphone and ask for questions after. So the floor is yours.
Thank you, Christian, for the nice introduction. You are all invited to the concert, the 21st of May. But today I'm going to speak about talk entitled Vaccine-like alternative method for plant protection, stimulating the innate immune system of plants. Doesn't work. Yes. I would like to have the pointer here. Okay. Contrary to animals, plants are sessile organisms. So they are not able to escape from a, let's say, stressful environment condition, and they are constantly exposed to a wide range of stress, which can be biotic or abiotic stress. Just to be clearer, in plant biology we are talking about biotic stress when a stress is induced by a living organism. It is the case of insect, fungi, infection of bacteria, etc. Or we are talking about abiotic stress when the stress is induced by environmental constraints. It is the case of high temperature, drought, light, and nutrient deficiency. Plants can experience stress in different parts of the plant. In the rhizosphere, which is the part of it's the soil, the part of the soil in which the root system is contained, or in the phyllosphere, which is the above ground part of the plant. So today I'm going to talk about phytopathogen, plant pathogens, and these organisms are defined as infectious agents that make plants sick. So it is known that plants pathogen can cause a percent of between 10 to 30% of the yield loss annually worldwide. Plants, when interact with pathogen, can massively respond at prescriptional level. And actually, it is known that up to 25% of the plant genes can respond to pathogen infections. It is nice to highlight that the disease is the exception, is not the rule, even because plants are constantly exposed to a wide range of pathogens, but most plants are resistant to most pathogens. And contrary to animals, plants don't have a circulatory immune system, but each cell is equipped to recognize pathogen and to trigger an immune response. So, but what makes an interaction into a disease? So, as I said, the disease it's not the role, but only a minimum part of the interaction, plant-pathogen interaction, lead to a disease. And this is why this is well represented by this model. It's quite known. It's defined as the disease triangle. And the disease triangle involved three different components. On one side, there is a pathogen that has to be virulent enough to avoid and to overcome the plant defenses. The host has to be susceptible to determine the pathogen, and there is also on the third side of the triangle there is the effect induced by the environment, and the environment has to be favorable for the infection. I could do a talk only about the effect of the potential effect of the environment on the plant pathogen interaction, but today I don't have time. And but if you are interested, I can invite you to have a look on a recent publication on a plant on the Journal of Experimental Botany, in which I've tried to summarize and to discuss the impact of different abiotic stress, environmental constraints, on the plant immune system, and also on the virulence of pathogens. Okay, but pathogen can utilize different strategies of pathogenicity. First of all, they have to find a determined host, and they have to attach to it. They can move passively or actively. For example, Botrytis cinerea can spread spores in the environment, which can be moved by wind, or other pathogen can be transported by insects. Otherwise, the passive movement can be through chemotaxis. Once a determined pathogen find a plant, has to overcome the physical barrier, and then has to also to avoid the plant defense system, has to grow and to reproduce inside the plants, and at the end has to spread to neighboring plants. So to enter, to penetrate the physical barriers, plant pathogen can utilize, in this case, a passive mechanism. For example, they can enter through wound, or they can also enter through a natural opening, such as a stomata or edotode, which are pores that are used by plants to exchange gases or water. Or they can use specialized organs, such as a pressorium, which is an organ which can press, we can lead to high pressure on the cell wall, creating pores. Or they can also use specialized enzymes that are defined as a cell wall degrading enzyme, and I'm going to talk about them later in the talk. So based on the lifestyle established by pathogen, we can group them in three different groups, big families, let's say, that are defined as a necrotroph, biotroph, and the hemibiotroph. So the necrotroph can establish with a plant an interaction which is quite devastating, because they need to grow, they need to kill plants, and they need to consume. Let's say they need to kill the plants and to consume dead tissue. The biotroph pathogen, they require the host to be alive, So they are used to establish a very complex genetics interaction with the host. And the last family group, it's defined as hemibiotroph, and this family it's a hybrid on the two previous groups. And so generally, this kind of pathogen can establish a biotrophic interaction during the first part of the life cycle, and then they can turn in necrotrophic during the second part of the lifestyle. Okay, but how does the plant immune system works? The plant immune system is quite intricate because, as I said to you before, each plant cell is equipped to recognize pathogen and to trigger an immune response. The plant immune system is made up of three different layers of defenses, defined as PTI, EPI, or SIR. PTI is acronyms of pattern trigger immunity. EPI is acronyms of effector trigger immunity, and SIR is a systemic acquired resistance. So EPI and PTI are triggered at the site of the infection and are triggered following perception of structure, microbial structures extracellularly, or are triggered following the intracellular perception of virulent factor. Once these two defense layers are induced, plants can diffuse the signaling molecules around plants in distal part, and in such a way they can trigger a systemic acquired resistance. To go a little bit more into the detail about how pathogens are recognized by plants, I have to introduce you the pattern recognition receptors. Plants actually feature hundreds of PRR, which are receptors made by two different parts, an extracellular part which is enriched in leucine repeat domain. This extracellular domain can recognize a bacterial pathogen, such as flagellin is the most known, or they can also recognize cell wall fragments generated by cell wall damages. So once there is the recognition of the pathogen of the damage, cell wall damage, this receptor can transduce the signal by using an intracellular domain, kinesis domain, which is going to transfer the signal to the nucleus. Okay, but nowadays it is known that upon perception of these patterns, plants can trigger temporal dynamics responses, and actually it is known that plants can be defenses can be triggered as early as 1 minute to 5. This is the case of the PRR complex formation, the phosphorylation of receptor kinases, or the accumulation of calcium, or the activation of ROS. Or they can be induced as late as 10 minutes to 2 days. This is the case of phosphorylation of MAP kinases, the accumulation of emanation of ethylene, defense gene expression, and stomatal closure to callus deposition. All these responses culminate in the PTI or DTI response. Okay, today I'm going to talk about the plant cell. Contrary to the animal cells, all the plant cells are surrounded by a barrier, which is defined as a plant cell. I don't want to enter and to discuss today about the chemistry of the plant cell wall, even because during my previous talk in the workshop of biostimulation and plant protection, I entered into the detail of the chemistry of the plant cell wall. But today I just want to say that the plant cell wall is made up of 2 or 3 different layers. As we can see, we can recognize the middle lamella, which is mainly enriched in pectin. The primary wall is enriched in hemicellulose, and in cellulose, and some cell type, plant cell type, are also characterized by secondary cell wall, which is rich in lignin. So historically, the plant cell wall has been always considered to confer a passive to carry out a passive defense barrier, let's say, for a pathogen to counteract a pathogen invasion. But on the other hand, in the last decades, several research shown that the plant cell wall can also represent a reservoir of signaling molecules, which can be recognized by plants and play a role in immunity. In fact, as I said previously, pathogens possess a very big arsenal of cell wall degrading enzymes, such as cellulase, pectinase, selenase, which aim to deconstruct the polysaccharides constituting the cell wall. So in turn, these enzymes can release inside the apoplast a blend of different oligosaccharides. Here I've reported cellobiose, other oligomers, and so on. So if plants can possess a specific cell wall localized receptor, and if they can recognize these sugars, they can also activate plant defenses. So during my research here at the ULB, I'm particularly interested in the degradation of cellulose. Nowadays, it is known that cellulose can be degraded by two different mechanisms: hydrolysis or oxidation. The main hydrolytic enzymes are endoglucanases, which can open the entry site of morphocellulose. The cellobiohydrolases can convert cellulose to cellobiose, and the beta-glucosidases that can convert cellobiose into glucose. On the other hand, quite recently, in 2010, the LPMO, the lytic polysaccharide monoxygenases, have been discovered, and this discovered, let's say, overturned the field of the cell wall research, because this enzyme can perform an oxidative cleavage of the cellulose and other polymers. In fact, LPMO are oxidative enzymes that can cleavage not only cellulose, but even other recalcitrant polysaccharides. Nowadays, the LPMOs are classified in 7 different classes based on the substrate specificity. These classes are defined as A9 to A11 or to A13 to A16. So in the presence of H2O2 or molecular oxygen, and in the presence of a reduced agent or in the presence of an enzymatic partner, such as cellobiose dehydrogenases, LPMO can oxidative cleaver the cellulose, which in turn release a blend of native and oxidized and sugar oxidized in the position C1 or C4 of the pyranose ring. Here on the right, I've reported a general chromatogram of the product of the LPMO acting on cellulose. So these enzymes are widely conserved among fungi and bacteria, and they are particularly enriched in phytopathogen. In fact, as you can see in the green box, in the gray box, Manioporta oryzae, Botrytis cinerea, and Fusarium graminearum, which are 3 very important phytopathogens, feature multiple copies of LPMOs of the class A9 in their genomes. Interestingly, Blanc Ulate in 2014, they studies the interaction between Botrytis cinerea and different hosts, such as lettuce, tomato, and grape. And they've demonstrated that the different LPMOs are used, are deployed by Botrytis gene area differently based on the host, which is infected. And I've started my research here by studying the expression of all the genes, of the LPMO genes of Botrytis gene area during the interaction with Arabidopsis. And similarly, I found that these genes were differently modulated, and I identified one which was highly modulated 45 hours after infection. So coming back on the activation of the immune system, I've decided to perform the different experimental processes. First of all, I've decided to work with the model plant Arabidopsis thaliana, because it's a model plant for molecular biology studies. It's characterized by having a short generation time. It has a small genome, which is completely sequenced and known, and it's easy to work with it actually. So I've treated the plants with the products of the LPMO and with CelloBIOS, which is as a positive control. CelloBIOS is a native cell-oligosaccharides, which was previously shown to have some elicitor properties on the immunity. Therefore, I've performed plant pathogen interaction studies, transcriptome analysis, hormonal and metabolite profiling, and several bio and histochemical assays. So, starting from the microscopic, let's say, symptoms, I've observed that pre-treating plants 24 hours before the infection with Botrytis, with the product of LPMO, observed a significant decrease of the 30% in the necrotic lesion area in Arabidopsis, and also in tomato. Actually, in tomato, the protection was even higher, because I observed a 70% reduction of in the necrosis area. So, to understand what happened after the treatment with the product of LPMOs, I performed transcriptomic analysis 1 hour after the treatment. And as you can see, I've identified almost a double number of upper-regulated genes following treatment with A9 COS as compared to CelloBIOS. I performed a PCA analysis, and actually the different samples were well separated, explaining almost 52% of the variance. And then I performed also a genontology analysis by using the 492 upper-regulated genes. And as you can see, I've identified 10 different clusters which were strongly enriched in the immune responses, such as innate immune regulation, cell wall modulation, signaling, and etc. Then, to understand the different responses induced by the oxidized cell-oligosaccharide compared to the native one, I performed a pairwise analysis by doing hierarchical clustering. And in this case, I have identified 10 different clusters. And then I perform, for each cluster, I perform a geo analysis, and I was interested in understanding the geotherm defined as defense responses. And as you can see from this graph, the first 3 clusters were enriched in genes belonging to this geotherm, indicating that the oxidized sugar can induce a stronger immune response as compared to the CelloBIOS, the native one. And then here you can see a neat map of several, let's say, key regulatory genes extracted from the 3 first cluster. And as you can see, I've identified the CIF2 and CIF4, which are 2 malectin-like receptor. FRK1 It's a kinesis which is induced by flagellin. MIB51, it's a transcription factor regulating glucosinolate, and WORK33, it's another key transcription factor regulating different aspects of immunity. So generally, when there is a recognition of flagellin or recognition of pattern in general, this recognition can induce an activation of reactive oxygen species. So I performed different biohistochemical assays to evaluate the production of ROS, and interestingly, both in the short term after the application of A9 COS and both later at 24 hours after treatment, I didn't observe any formation of ROS. This is quite interesting. Then I focused my attention on the phytohormones. Phytohormones can play key roles in activating the plant immune system. In particular, the three main hormones regulating different aspects of plant defenses are salicylic acid, osmonic acid, and ethylene. Therefore, I've quantified osmonic acid and salicylic acid 24 hours after treatment with A9 COS. And as you can see, I've detected a significant greater amount of these two hormones. And then I performed also a time course analysis, starting from 2 to 24 hours after treatment to evaluate the emanation of ethylene. And interestingly, identified and I observed a significant accumulation of this hormone, starting from 2 till 4 hours after treatment. Okay. Then, from the transcriptomic analysis, I also identified several other receptor kinases, RLKs, which are well known to be involved in signaling the plant defense responses. And I've selected some of them. In particular, I've selected the BAC1. BAC1, it's a co-receptor of two well-known receptor kinases, such as FLS2 and EFR, which are specific receptor for flagellin and elongation factor. I've selected TZOS1, T1, which is another receptor which is used by plants to monitor the alteration of the cell wall. And so it's a kind of a sensor of the cell wall integrity. And then I've selected other two receptor, malectin-like receptor, defined as a stress-induced factor 2 and 4, which were been recently shown to be involved in stomata immunity. So to study the involvement of these different components, I've performed gene expression analysis by evaluating 4 or 5 marker genes in wild-type plants, and in single mutants. And as you can see, in all the single mutants, the defense gene expression was impaired, indicating that all these components are required to fully signal the A9-dependent defensive responses. Then I focused my attention on camalexin. Camalexin, it's antimicrobial compounds belonging to the class of the phytoalexin. Nowadays, the signaling behind the production of camalexin is well known, and it involves the phosphorylation of MAP kinases, which in turn is going to activate the WORK33 transcription factor, and which is going to regulate different camalexin biosynthetic genes and transporter. So I performed a Western blot analysis 5 minutes after treatment with the cellobiose and with the products of LPMO. And interestingly, I identified the phosphorylation of both the MAP kinases 3 and 6 only after treatment with the A9 COS. Then I've performed gene expression analysis for several genes involved in the signaling of the camalexin biosynthesis 1 hour after treatment. And analogously, I observed a significantly expression only after treatment with A9 COS. And finally, I quantified camalexin 24 hours after treatment, and even in this case, camalexin accumulation was significantly higher only after treatment with A9 COS. So then I was wondering if the two malectin-like receptors, CIF2 and 4, were involved in signaling the camalexin production. And actually, I quantified camalexin following treatment with cellobiose and A9 COS in both loss-of-function mutants. And interestingly, we didn't see any accumulation of camalexin. On the other hand, I performed a Western blot in the 5 minutes after treatment in the two loss-of-function mutants, and interestingly, I observed a phosphorylation of these MAP kinases. Therefore, I proposed a little, let's say, schema to describe the camalexin, the way of the signaling behind the camalexin production induced by A9 COS. And to have the production of camalexin, we need functional CIF2 and 4, because probably these two receptor kinases can block the synthesis of camalexin at the level of WRT33. So altogether, I can say I proposed, let's say, a schema in which I've hypothesized that the activity of LPMO on cellulose can release inside the apoplast a blend of native oxidized cellulose oligosaccharides, which can be perceived by receptor-associated cell wall receptor kinases. We are working to identify the specific receptor, but for sure I can say that T-Zeus, BAC1, CIF2, 4 are required to signaling the defense responses, which can lead to the camalexin synthesis. It can lead to phytohormones accumulation, callus deposition, and from the transcriptomic analysis, we also saw that even the sugar importer and exporter are modulated. So, to conclude, as take-home messages, I can say that the oxidation of cellulose can play a key role in immunity, in plant immunity. The A9 COS can be considered as vaccine-like molecules, since they can act as damage-associated molecular pattern. This molecule may also signal the cell wall integrity during both abiotic and abiotic stress, and surely with the discovery of LPMO open a bigger perspective on the field of the sweet immunity, possibly defined as oxidized sweet immunity. I would like to thank all the group that it's hosting me, the CPPL group, and I would like to thank also Professor Cédric Delporte for help in quantifying the camalexin. The two colleagues from the University of Regensburg for the transcriptomic analysis, and several collaborators at the IGPB ERA in Bruxelles for help and discussion. Thank you for the attention.
The questions. Loic first.
Okay, just a little question about the treatment that you applied. So you applied LPMO and cellulobiose like separately?
Yes, separately. The product of LPMO without LPMO. So the sugar deriving from the reaction of LPMO on cellulose.
I was not sure to understand the motivation of just putting cellulobiose without.
Because inside the blend of the sugars generated by LPMO, okay, there is also a little part of cellulobiose, but LPMO can perform an oxidative, let's say, breakdown of the cellulose. They can generate a part of native and a part of oxidized sugars. So since we were interested in understanding the oxidized sugar, the role played by the oxidized sugars present in this blend, we use it as a control cellulobiose, and to do a comparison, okay? Because cellulobiose is not oxidized.
So it was considered as the control treatment?
Yes, we use it as a control, yes.
Okay. Thank you.
Mark.
Yeah, very nice presentation. It's not my expertise, but I enjoyed the talk.
Thank you.
Just for my understanding, this AA9 cost that is normally made by botrytis, or not?
Yes, sir.
Yeah. So this is part of the natural defense against botrytis.
No,
So the
no.
plant recognizes this, and then it would be able to defend itself against botrytis. And then it doesn't work always, so apparently botrytis is quicker, and then it's able to infect plants despite the response.
Yes.
Right.
Mm-hmm.
And if you apply AA9 cost, then do you actually prohibit botrytis to infect, or do you only delay botrytis to infect?
Yes, actually the symptomatology studies that we performed was 5 days after botrytis infection, and actually we didn't wait longer, and probably we have to check if there is a delay or it's blocked. We still don't know.
Okay.
But I can say that I've created some, I didn't talk about it, but I created some CRISPR-Cas9 strain of botrytis without an LPMO, and it's significantly less virulent.
Yeah, okay. But it is, so because you also claim in the title that you want to use it as a vaccine,
Yes.
and then it has to be working perfectly, of course.
Yes, yes.
So I was just wondering how perfectly it is working, and whether you have any evidence.
It works, yes, it works at least, we know that at least after 5 days it's effective.
Okay.
Then, yes, on the field, maybe it's required to do, I don't know, several treatments, probably a booster, yes, 3 or 4 times. And yes.
And then another thing, you only used Colombia to test this. Have you tried other accessions? Because I can imagine that there is a lot of variation.
Actually, no, only Colombia.
So it may actually be that there are different receptors that are acting a little bit faster or a little bit slower, or they look at different molecules.
Absolutely, yes, good idea.
And did you look at other botrytis strains? Because this was a strain which is genic on Arabidopsis as well.
Yes.
Yeah. And have you tried others or not?
No.
Okay. And any other pathogens that are also necrotrophic, that may use the same mechanism?
Necrotroph for now, but we try with Pseudomonas syringae, which is a biotroph. And actually, we saw a higher protection because the left side of the Pseudomonas is less devastating compared to botrytis. And yes, the treatment against Pseudomonas is even more efficient. Yes.
And the last question is, I can imagine that if you make molecules that are overly sensitive for this particular signal, or that are more or less constitutively signaling, then the plants would also be more protected against the pathogen. But what would be the effect? Would it also give a very strong effect on growth?
And the growth, yes, we are working on it because actually these are sugars and can also be good to induce the growth. Because actually the effect
I mean the growth of the plant, not the growth
of the plant, yes.
Yeah.
But yes, the risk is like, kind of no, it's not a dogma, but it's an open question about the trade-off of defense and the growth of plants. But actually, the defense responses here are maintained at least at molecular level till 3-4 hours, are not very long at molecular level.
Yeah.
And we are working on it because apparently we can also stimulate the growth, and this could be amazing.
Yeah. Yeah. Okay. Thanks.
Thank you.
Did you try just giving the LPMOs directly to the botrytis?
On botrytis?
Because you're initially like you're spraying it on the plants so that they're ready for the infection, and then you're applying the infection.
Yes. Yes.
Yeah.
No, directly on botrytis, no.
Just to see the effect on both the sides, like host and pathogen.
Yes.
I didn't get it. The enzyme or the sugars?
Enzyme.
Inside.
Yes.
What is already producing?
I'm sorry.
The drug is already producing the
Other questions on workies. So did you try on the working mutants as well? Because 22 and 33 are known for innate immunity, well established ones.
Yes, no, I haven't tried. Actually, should work.
Because you also should get the double mutant 22, 33.
Yes.
There should be double mutant as well.
Yes. Actually, yeah, you're right. Would be interested also in some amoeba.
Because you really have the chasmatins, like the cell selection, like
Yes.
probably like you'll be able to trace out the pathway.
Yeah. From the gene expression analysis, we showed that the CIP2 and CIP4 are blocking the expression of WORK33, but not the phosphorylation of MAP kinase. So probably yes, WORK33 is a key regulatory point. And actually, yes, it could be nice to work with the mutant plants. Yes. Good idea.
Actually, I would like to add generically that this was the first kind of this work based on the cellulose, and then was like, let's say, very preliminary. So that if you compare this one to the oligosaccharides, the level of work that we've done in the last 2 decades, I would say these are completely new class of molecules, this oxidized cellulose, that I think it deserves like at least minimum 5, 6 or a decade, or 5, 6 years or a decade, just to have a bit bigger picture than what we already have with the active level of mutagens. So it's very preliminary. So majority of all these questions I heard today are actually all those super valid questions that not only Marco can, I mean, many other people are now starting to work on this through collaboration with us or even independently in their own labs. So I'm really confident that this will be like a launching on new field, this oxidized weak immunity.
The question online, you can unmute under the microphone. I see Michaela is online. No question? No. In the room?
No.
Thanks. Thanks for the presentation. Very nice. As Mark is not my expertise, but you just checked this response using the oxidized sugars.
Yes.
So, do you think maybe any oxidized product can trigger this kind of effect? It's not for the sugars, but the oxidized terminal, because you have a pool from C2 up to C6 maybe or more, and you use just cell.
Yes.
And then the effect is from the oxidized terminal from the sugar, right? Not
yes.
from this sugar. Maybe if you use other oxidizing products, can this oxidizing product trigger the same effect, or
Other product? Do you mean other sugars or other?
Organic acid in general.
or
Organic
Like a
acid.
C4, I mean like
Okay.
butyric acid or anything.
Yes, I should try. I don't know.
no, because,
Yes.
because during the infection process, fungi need to change the structure of the cell wall, and generally they start with oxidative enzymes. Can be
Yes.
AA or can be any AA.
Oh yes, yes, for sure. Yes, it can be. It can be. I'm quite confident it can be. Then it. Yes, it's a good question.
Yes, as I told you, it's not my
Yes,
my
but probably.
expertise, but I was just
Actually, we saw that the oxidation of cellulose, probably pectin, even lignin, is possible. Is perceived by plants.
maybe even if you add oxidizing chitin,
Yes,
maybe can trigger some response.
even because the cell wall integrity is, besides the immunity, there is also a big, let's say, plants can invest a lot of energy in monitoring the cell wall integrity, and I'm quite confident that they can recognize oxidation, which is which is an important phenomenon.
Yeah.
And yes. But the cell wall integrity responses are a little bit different compared to the immunity response. They can be overlapped a bit, but they are a bit different. Yes.
Marco, you come from a previous experience on vegan acid, remember?
Biacid.
Yeah, we were working on vegan acid.
Yes.
And you get a mixture of any kind of acids possible coming from organic acid.
Yes, actually.
You can talk about that.
Yes, it's previously characterized the immune response induced by bile acid. In particular, it was a secondary bile acid, the oxycholic acid, and which can also be oxidized, I guess.
Yeah.
Yeah.
Libre do that.
Yeah.
A lot of them.
Yeah. Yes. But here it's nice because the oxidation can from inside the plant.
Okay.
But maybe yes, it can work.
Thanks.
Thank you for the question. Nice.
No, thank you very much.
Are there more questions? Then with this we can close with the that you have extra announcement.
Well, first, just thanks again, Marco, for a really great talk. No, it's a little bit bittersweet because Marco gave our last BrIAS Talks. So thanks so much for that. A little bit teared up there. Tomorrow we have another workshop starting at 1 p.m. in the South Solvay. It is, let me bring up the title. It's on the long-term influence of imperialism, colonialism, acculturation on agricultural and dietary change in Africa. And here we are welcoming historians and archaeologists specialized in Sudan, Eritrea, Tunisia, Roman Britain, and West Africa. So really a lot of interesting topics. You don't want to miss it. You can join us online and in South Solvay. And Thursday, of course, the 14th, we have our final event, a closing event of our program, where we will hear talks by many of our team members summarizing what we all accomplished together during our spring program. And we are also welcoming two guest speakers from the FAO, Mr. Rashad Al-Khafaji, the director of the FAO liaison office with the European Union and Belgium, and our BrIAS fellow coming back to us, Dr. Ratana Norbert Mons, based at Utrecht, and the FAO Regional Office of Asia and the Pacific. So please join us in the South Solvay or online. Following our closing event, we have a drinks reception which you are welcome to join us at. And of course, we welcome you to follow BrIAS and more about our upcoming activities in the future on our website, LinkedIn, Facebook, etc. And YouTube. I am busy uploading more videos, so please to reflect on the wonderful program we have and revisit some of your favorite events. Please have a look at our YouTube channel and subscribe. So thanks very much.